Hello, young biologists. Have you ever wondered how do we know that plants actually absorb water from the soil and send it all the way to their leaves? We can't see it happening with our eyes, right? That's where simple experiments come to our rescue. Through clever setups using test tubes, dyes, and live plants, we can make the invisible journey of water visible. In this session, let's explore a series of fascinating experiments that prove how roots absorb water and how xylem tissues transport it upward, keeping plants alive and thriving. Let's begin with our first experiment. Imagine pulling out a small balsam plant from the soil and placing its roots carefully into a test tube filled with water. To make sure evaporation doesn't confuse our results, we add a thin layer of oil on top. Now, what do we observe? After a day or two, the water level in this test tube falls, while in a controlled test tube without a plant, the water level remains unchanged. The conclusion is crystal clear: roots are actively absorbing water from their surroundings. Isn't it amazing how such a simple setup can prove a fundamental life process? Now let's see how that absorbed water moves upward. In this experiment, a young balsam plant is placed in a beaker containing eosin solution, a pink dye. After a few hours, if we slice the root, stem, and leaf and examine them, we see beautiful red stained lines in the veins. These stained parts are none other than the xylem vessels. The dye traveled with the absorbed water, moving upwards through xylem. This gives us visible proof that xylem tissues act like water pipes in plants, carrying life-giving water all the way to the top. To confirm that water really moves only through xylem, scientists devised another experiment. Take two leafy shoots. In one, remove only the outer phloem ring, keeping the xylem intact. In the second, remove the central xylem, leaving only phloem. What happens? The shoot with intact xylem remains fresh and turgid, but the shoot without xylem wilts and droops. This clearly shows that phloem cannot transport water, only xylem does the job. Nature has perfectly designed tissues for different purposes: xylem for water, phloem for food. Here comes the classic ringing experiment. In a woody stem, a ring of bark, which contains the phloem, is carefully removed while the inner xylem is left intact. After some weeks, the stem above the ring swells because food manufactured in leaves accumulates there, unable to move down. Meanwhile, the lower part of the stem may shrink or even die. This experiment tells us two important things: water moves upward through xylem, while food moves downward through phloem. Together, these two tissues form a perfect transport system that keeps plants alive and balanced. So, let's quickly recap. We saw that roots absorb water directly from the soil. Staining experiments with dyes gave us visible proof of water's upward movement through xylem. Comparative twig experiments confirmed that xylem is the sole pathway for water transport. And finally, the ringing experiment showed us that phloem carries food, not water. Isn't it incredible how simple classroom experiments can reveal such complex plant physiology? Seedling every glass of water you pour into the soil is actually fueling a silent, upward journey inside the plant. I hope this session has helped you appreciate the elegance of these processes. Until next time, keep observing the living world around you, because biology is happening all the time, right before your eyes.